The overall goal of this procedure is to purify vesicles from blood while reducing co-purifying proteins and complexes which can interfere with downstream analyses, including nanoparticle tracking and proteomics.
This method can help answer key questions such as what are the proteomic changes in serum or plasma vesicles during a disease state as compared to a healthy state. The main advantage of this technique is the increased accuracy in vesicle enumeration. In removing abundant non-vesicle contaminants, we increase the identification of low-abundance proteins and the potential biomarkers of disease.
The implication of this method extends toward the discovery of novel diagnostic biomarkers. It enables more accurate determination of the proteomic composition of extracellular vesicles in complex matrices, like blood.
Generally, individuals new to vesicle purification will struggle because there are many published techniques. The reproducibility of downstream proteomic analyses is affected by the co-purification of abundant serum proteins.
Equilibrate a serum or plasma sample by placing the tube under non-shaking conditions in a 4°C refrigerator overnight. The next day, aliquot 250 microliters of the sample into a microcentrifuge tube using a 1000 microliter pipette. Centrifuge the sample at 18,000 times G for 30 minutes at 4°C. Using a 200 microliter pipette, remove 200 microliters of the cleared supernatant and add it to a new microcentrifuge tube. Avoid any pelleted material while transferring the supernatant. Quantify the protein content of the sample by BCA assay using the manufacturer's protocol. To perform the assay, dilute the sample 1:50 in 1x phosphate buffered saline.